is Suzanne, and I work as a research assistant at the National Horizon Center, Teesside University. I also did my master's degree in the university. I'll talk about Lewy body dementia. It's the second most common cause of dementia after Alzheimer's disease. The Lewy body dementia is a spectrum disease, covers a couple of diseases: dementia with Lewy bodies, Parkinson's disease, and Parkinson's disease dementia. Parkinson's disease dementia. Dementia with Lewy body is often called as the most common disease you've never heard of. I've never heard of the disease until my father-in-law was diagnosed with it, and public awareness grew as Robin Williams actually was diagnosed with the disease after his suicide. Parkinson's disease, we all know Michael J. Fox, who starred the film Back to the Future, and he was diagnosed with the disease at the age of 29. 10 years later, he started his foundation to support Parkinson's research. Just last week, President Biden signed the National Plan to End Parkinson's Act into a law, an initiative we wish to see similar in the UK. Lewy body dementia pathology. As you all know, that Lewy body is the main pathological characteristic of these diseases. It consists of mainly alpha-synuclein protein. However, recent imaging techniques showed that Lewy bodies can consist of mitochondria, lysosomes, and lipid-rich vesicles. And so the aggregation of alpha-synuclein is intrinsically disordered monomer, so it's in the brain, in the cytoplasm of the neurons as monomer, and it can be bound to the membranes as well. That's in the physiological state. However, in the pathological state, alpha-synuclein aggregates and form oligomers and fibrils, and these are toxic. The video shows how the fibrils accumulate and spread in the neurons. However, in the literature, we found that these oligomers and fibrils are toxic. However, recent research shows that after the aggregation, phosphorylation of alpha-synuclein at serine 129 might have a protective aspect to the neurons. In our research, we try to investigate this aggregation process, as every step might have a diagnostic aspect to it. So we use cellular models to aggregate alpha-synuclein. We produce and purify alpha-synuclein in the lab, and then seed the cells, in genetically engineered cells, to overexpress alpha-synuclein. We seed them with preformed fibrils. We aggregate the fibrils as well in the lab, add the aggregates, the fibrils, to the cell lines, and then with Raman spectroscopy, with Raman spectroscopy, we detect the changes in the cells over 14 days. Raman spectroscopy is a non-destructive technique, label-free, and it gives a unique fingerprint of the components of the sample. We use a Raman shine the laser. It's a laser-based technique. We shine the laser on the sample, and it gives us a spectrum, and each peak of the spectrum corresponds to a biomolecule. We then use a library to assign these peaks to the biomolecules and detect the changes of the sample. So the main results of all peaks that Raman have given us, we see significant reductions in the phenylalanine and the glycosidic bonds. We also see reductions in sphingomyelin, fatty acids, and liposomes, where the PCA plots of overall Raman was able to delineate between the treated groups of cells and non-treated groups. The treated groups are treated with PFFs, and non-treated groups and control groups. And we also used Raman to detect the changes in the CSF of PDD and DLB samples. And it was successful in delineation between the disease groups and control groups. There were overlapping between the disease group PDD and DLB. In this, as a summary, Raman spectroscopy proved to be a sensitive technique to differentiate between cells with PFFs and cells without PFFs, and in the CSF it was also sensitive to delineate between the control group and the disease groups. The changes in the lipid profiles may indicate a disruption in the cellular membranes and the cellular processes. And most spectral changes seen early in day one during the aggregation process, and the changes were prominent in the cell models in the HEC293 cells, and was similar and less pronounced in other cell lines. And that's all for me. Thank you. Thank you.